Hello everyone, welcome to ITLS Academy. In this session, we will be covering part 2 of the topic Fundamental Concepts in Immunology. I am your mentor, Dr. Shamaita Basu. So, last session means the part 1 of this topic was ended with the innate immunity. So, in this session, we will start with the adaptive immunity. Okay. So, some of the important features of the adaptive immunity will be that it is specific, it is the second line of defense, and it is having a memory. Means if there is repeated exposure of the same pathogen, then by virtue of the adaptive immunity, your body will be able to fight against that same infection for the second time, which was not there in case of innate immunity because it was a non-specific line of defense. Okay. It is also having a faster response, a more vigorous response, means a more strong and bold response, and also a long lasting response, obviously, because the memory cells are there. Okay, so obviously the response will be long lasting, which was also not there in case of innate immunity. And it is also anamnestic. So anamnestic means it is the it is that type of the immune system which is responsible or which can be helpful in the process of vaccine production, because the entire concept of vaccine production is based on the concept of adaptive immunity. So hence it's an anamnestic response also. Now talking about the components of the adaptive immunity, again very important. So we have the cell mediated component, or we have the cell mediated part, and we also have the humoral part. Okay. So the cell mediated part will basically involve the T cells. We will be coming to it, and the humoral part or the humoral immunity will mainly involve the antibodies, which are also the soluble factors. Now moving forward, what are the cardinal features, the specific important features of the adaptive immune response? Okay, so firstly, it will be specificity. So it is specific for distinct antigen, for different structural components of a single complex protein, polysaccharide, or other macromolecules. There are portions of such antigens which are recognized by individual lymphocytes, and these are called the determinants of the epitopes. Epitopes are again a very important concept when we are talking about adaptive immunity. I will be explaining to you with a nice diagram what are epitopes. And this fine specificity exists because individual lymphocyte express membrane receptors able to distinguish the slight differences in structure between distinct antigens, and that is where the importance of epitopes comes in. Okay, because without epitopes the antibodies will not be able to recognize the minor differences that are there between the antigens, because as we also saw in case of COVID pandemic, that rapidly new strains or rapidly new variants of COVID were coming up. Why? Because the COVID virus was constantly evolving, it was constantly mutating, it was constantly changing itself. Okay, now changing in what manner? Changing in what manner? It was changing the antigenic properties. Okay, so some of the antigens, because what are antigens? Antigens are nothing but proteins. And what are proteins? Proteins are nothing but groups of amino acids. So a slight change in a sequence or in a set of amino acids will actually lead to change of that particular protein. So these are slight changes which must be recognized by your immune system in order to be able to fight against that particular pathogen. And that is where the importance of the epitopes comes. Because if there is no particular specificity or no particular region by which you will go and recognize that particular antigen, then also when in future that antigen will mutate, that antigen will evolve and infect you for the second time, your body will not be able to recognize that okay, it's a new variant or it's a new type. Okay, so that is where the importance of epitope comes in. Now coming to the next important feature, that is memory. Again, a very very important feature of the adaptive immune system. So it is exposure of the immune system to foreign antigen which the body needs to remind, or which the body needs to remember about. So it enhances its ability to respond again to that same antigen, and responses to second and subsequent exposure to the same antigen, which is called secondary immune responses, are usually more rapid and larger than the first or primary immune response. This part is very important. Secondary immune response is much more stronger and effective and rapid than the primary immune response. Why? Because your body already knows what to do. isn't it? Just as like whenever we learn something new, it can be any type of activity, it can be any type of new topic, it can be any type of new course, or just any type of daily new core that we learn about. Okay, it can be as simple as cooking, or as simple as dancing, or like when a baby starts to walk, when a baby starts to learn to speak. Okay, so anything that we do for the first time, we are not confident, we are not experienced, we don't know how to do it. But that same thing, when we do for the second time or the third time or the fourth time, by that time we are very much confident. We are very much experienced. We know what to do and what not to do. We know how to do it and how not to do it, isn't it? So it's the same story with the immune system of our body also. So for the first time when the body is getting exposed to a particular antigen, the body is not prepared. The body doesn't know that how much harmful that pathogen is, how much cells, or how much effort the immune system needs to combat that particular antigen. Anything of it is not known. But when the body, or when the immune system is combating the same antigen for the second time, that time also obviously the body already knows, because it has produced the memory cells. The immune system has already produced the memory cells, so the memory cells will remind them. Okay, just as when we look at a person, or when we look at an object for the first time, it sets in our memory, isn't it? So when for the second time we again meet that same person, or when for the second time we again go to that same place, we can remind ourselves, ki, okay, this is that place, ki, okay, this is that person that I met that day over there. Okay, so that is exactly the job of memory, or that is exactly the job of memory cells of the immune system, to remind the immune system that you already know this antigen, you are already prepared to fight against this antigen. So that is the boost, that is the confidence that the body gets, and hence the secondary line, or sorry, the secondary immune response is much more effective and long lasting. Okay, now coming to something called specialization. So the immune system responds in distinct and special ways to different microbes, maximizing the efficiency of antimicrobial defense mechanisms. Thus, humoral immunity and cell-mediated immunity are elicited by different classes of microbes, or by the same microbes at different stages of infection. It can be extracellular or intracellular, because some microbes are there. Mainly this extracellular and intracellular concept is like applicable for the bacteria, because some bacteria are there which infect extracellularly, and some bacteria are there that infect intracellularly. Okay, so depending on that, specialization is there, okay, which actually maximizes the efficiency of the antimicrobial defense mechanism. Okay, and what is about self limitation? So all the normal immune responses returning the immune system to its resting or basal state with time after antigen stimulation, this process is called homeostasis. Now basically, what is homeostasis? Homeostasis is normal functioning of the body. Okay, normal functioning of the body, which also includes tackling any kind of infectious situation, or tackling any kind of hazardous or harmful situation. Okay, so it is like whenever the body is exposed to any kind of infection, it is exposed to any kind of infectious pathogen, there is a rise in the temperature, there is a change in the pH, normal pH of the body, which is usually between 7.2 to 7.4. So these are some of the changes that needs to happen when your body is under threat. But these are also some of the changes that needs to be rectified at the same point of time. So that is what is homeostasis. So if this homeostasis or self-limitation is not regulated, it is not controlled, then what will happen? Then the overall functioning of the body, the overall normal physiology of the body will be hampered, which actually happens in case of cancer, because cancer is a situation where your own body is not in your favor. Okay, where your own body cells have started multiplying in a rampant way, in an uncontrolled manner, in an uninformed and unplanned manner. Okay, so that is a situation where the entire homeostasis of the body is gone, the entire homeostasis of the body has gone haywire. Okay, so that is the situation when we say that our body has lost it. Okay, so homeostasis is something very important for the normal physiology or the normal functioning of the body. Moving forward, an effective immune response will involve three major groups of cells. Very important. What are they? The cellular immunity, which will involve the T lymphocytes or the T cells, because lymphocytes are of two types. What are lymphocytes? It's a type of WBC. So it's of two types. One are the T cells and the other one are the B cells. Okay. So they are also known as T lymphocytes and B lymphocytes. And we have the humoral immunity, which will involve the B cells or the B lymphocytes. And we also have the accessory cells, which are the antigen presenting cells, or they are also known as the T helper cells. Okay. So antigen presenting cells are actually also represented as APCs, short form. Okay. So the two major population of lymphocytes are the B lymphocytes and the T lymphocytes, which provide us with our specific adaptive immune response. Okay. So now coming to the one of the major important concepts of adaptive immunity, that is antigen antibody. So what is an antigen? So it is a substance that elicits an immune response, which is obviously either an antibody response or a cell mediated immune response. So now not only a substance, I would like to specify over here a protein substance. Okay, because whether an antigen or an antibody, both of it are nothing but proteins in nature. Chemical nature will be protein. Okay, no matter what type of antigen or antibody it is. Okay. So basically the protein which is responsible for eliciting or responsible for inducing an immune response, that is an antigen. Okay. And antibody is what? Antibody is the protein which is produced in response to a antigen. Okay. So if there is no antigen, there will be no antibody. So they are actually complementary to each other. Okay. Now, what is the epitope? Okay, the important part that I was talking about in the previous slide, that is the antigenic determinant. Epitope is nothing but antigenic determinant. So it is that portion of an antigen which is recognized and bound by an antibody or a T cell receptor, that is the TCR. T cell receptor is also abbreviated as TCR. So epitope is nothing but antigenic determinant. So this is the main concept that you need to know about. That is, epitope is antigenic determinant. Okay, and it is a particular region or a portion of an antigen which will be recognized by the antibody, and therefore the antibody will go and bind to that part of the antigen. Okay. So moving forward, this is actually a nice diagram of the epitope that I have given over here. Okay. Now one protein will have multiple antigenic determinants. That is also a scenario or a fact. Okay. So epitope is that portion of an antigen that is recognized and bound to by an antibody or a T cell receptor. So see, this is basically your antigen that you have. And these antigen is having, see, three epitopes. So to these three epitopes, three different antibodies can come and bind. Okay. To see, to these epitopes, the first epitope antibody A has come and bind. To the second epitope, antibody B has come and bind. And to the third epitope, antibody C has come and bind. So that means what? This is a single antigen. This is only one protein, but it is having three different antigenic determinants. So three different antibodies can come and bind with that particular antigen. Okay. So that is the main thing. And see, the configuration is also different, which is separately shown over here. So antibody A is binding to this type of epitope. Antigen B is binding to this type of the second type of epitope, and antigen C is binding to the third type of epitope. So see, the structural configuration is also different, but all these epitopes are present on the same antigen. So the moral of the story is one particular antigen can have several different epitopes or several different antigenic determinant sites. Okay. Now coming to the concept of haptens, very important. What are haptens? Haptens are also known as incomplete antibodies. Okay, sorry, not antibodies, incomplete antigens. So these are small molecules which are non-immunogenic, thus could never induce an immune response by themselves. So basically the concept of haptens is that they are never competent or self-sufficient to induce an immune response inside the body of an individual. Okay, but they are antigenic in nature. So that's why they are known as incomplete antigens. Okay, because if it's a complete antigen, then the antigen alone will be able to will be able to elicit it or able to induce an immune response. Okay, So incomplete antigen, why? Because it is not sufficient to elicit or to induce an immune response on its own. So that's why it is non-immunogenic. Understood? Uh, so see, that is what they have shown over here. Do not elicit an immune response by itself. Now, what is immunogenicity? It is the ability to induce a humoral or a cell-mediated immune response. Because here we are talking about adaptive immunity only. So, okay, adaptive or acquired immunity only. So there is no question of innate immune response. So here we are only focusing on adaptive or acquired immunity. Okay. So immunogenicity is basically the ability to induce a humoral antibody response or a cell-mediated immune response. So basically immunogenicity is the integral property of any particular antigen. Not hapten but antigen, because haptens are incomplete antigens. So basically haptens are acting as the carriers. Okay, they are acting as the carriers of the antigen, so they are just helping in the process. Okay, they are like kind of accessory cells. Now immunogens can either be weak or they can be strong. Okay, so weak immunogens means they will be eliciting a weak immune response, and strong immunogens means they will be eliciting a very strong or a long lasting immune response. Okay, whether it's cell mediated or humoral antibody mediated. Now what determines immunogenicity? Foreignness. Okay, foreignness means something which has come outside of your body, something which is not a part of your body. Again, a very important concept. Okay, why a very important concept? Because from this concept only the idea or the concept of autoimmune disease arises. Okay, what is autoimmune disease? Auto means self, means when your own body is producing immune response, when your own body is producing antibodies against your own cells. Okay, so say for example, my body has started producing antibodies against my liver. So what will happen? Liver means, what are the liver cells known as? They are known as the hepatocytes, isn't it? So what will happen? The antibodies that were produced by my body will go and start attacking the hepatocytes, and that will lead to damage of my liver. And liver is one of the main organs of the body. It is one of the vital organs of the body. So if the liver is damaged, if there is hepatic damage, that means that person is gone forever. Okay, so that is this is just a a hypothetical idea or a hypothetical situation that I'm explaining. Okay, so that is the main concept of autoimmune disease. So when this autoimmune disease will not occur, when our body is able to recognize between self and non-self. Self means what is my own, and non-self means what is not mine and what has come from outside. That means what is foreign. So the main concept of the immune system is that the immune systems, the immune cells should be able to recognize those pathogens which are coming from outside the body, and also recognize those tissues which are of our own, which are of our own body. And after recognizing these two, they will only elicit the response, or they will only fight, or they will only exert the defensive mechanisms against the non-self particles or against the foreign particles only. So when this barrier, when this ability to differentiate between self and non-self is gone, that is the situation when autoimmune diseases develops. Okay, so there are many different examples of autoimmune disease, like we have rheumatoid arthritis, we have myasthenia gravis, we have muscular dystrophy. These are all various types of autoimmune diseases. Size. Size is also very important. So whether antigen or antibody is bigger or smaller. Then chemical composition, whether it's made up of proteins or nucleic acids or polysaccharides or lipids. And structure. Structure may obviously primary, secondary or tertiary structure will play an important role. Tertiary structure will obviously be the most effective. Okay. And also physical form, means whether it's particulate or soluble, whether it's soluble or insoluble. Now what is an antibody? So antibody will be produced by our body, isn't it? Antibody will be produced by our body, and by what? By the plasma cells. and these plasma cells will be in turn produced by the B cells or the B lymphocytes. So whenever there is a situation, or whenever there is a need to develop an antibody, or to produce an antibody, the B lymphocytes will be informed by the T helper cells, and the B lymphocytes will start producing the plasma cells, and those plasma cells will start producing the antibodies in return. So that is the entire story, or the entire cascade. So antibody is obviously an essential part of adaptive immunity. So it will go and specifically bind a unique antigenic epitope, means antigenic determinant, about which I have already talked about in details, and it also possesses the antigen binding sites. Okay, so on antigen we will have the epitopes, means the antigenic determinants, and on antibody we will be having the antigen binding sites. So whenever there is an antigen antibody reaction, whenever the antigen and antibody are reacting with each other, the reaction is basically occurring between the epitope, means the antigenic determinant, and the antigen binding site on the antibody. Clear? And these antibodies are basically members of the class of proteins called immunoglobulins. Okay, so as I already mentioned, that antibodies are nothing but proteins. Okay, so all the plasma proteins that we have inside our body, plasma proteins means proteins present in our plasma. So there are different classes of plasma proteins. We have albumin, we have globulin. Okay, so among them, under the globulin class, we have these immunoglobulins, which are nothing but the antibodies. So you can say, you can remember it like this, that immunoglobulins are a good name for antibody. Okay. Now coming to the structure of antibody, very important concept when we are talking about adaptive immunity. Okay, so see, just focus on this diagram. So it's a Y-shaped structure, you can see, isn't it? It is just looking like a Y. Okay, so in this Y-shaped structure, what we have two identical heavy chains and two identical light sorry, two identical heavy chains and two identical light chains. So as we can see, the heavy chains are marked with violet and the light chains are marked with different shades of green. Okay, so the heavy chains are bigger. The Y-shaped structure is actually formed by the heavy chains, and the light chains are shorter. So each heavy chain and each light chain is in turn having a constant region and a variable region, which are in turn marked by the light and the deep shades. So the deep violet shades will be the heavy regions of the heavy chain, and variable regions of the heavy chain, and the deep green shades will be the variable regions of the light chains. Okay, so on the upper side basically we have the variable regions, and on the lower side of the Y chain, of the Y-shaped structure, we have the constant regions. Okay, so variable regions and constant regions are something common for both the heavy chains and the light chains. Okay, and these bridges, as we can see over here, see this red bridge over here, this red bridge over here, this red bridge, these are basically the disulfide linkages or the disulfide bonds, which are basically holding together the heavy chains and the light chains. So that is the major concept or the overall view of the antibody structure. Now, what is an important part of the antibody structure? That is the FAB part. Okay, what does FAB stands for? Fragment of antigen binding. So F stands for fragment and AB stands for antigen binding. Okay, so what is where this FAB part will be situated? It will be situated on the variable region of the both the heavy and the light chains. Okay, so see, on the top region means the variable region, and we have it both on the heavy chain as well as on the light chain. So these upper parts, this will be the antigen binding sites or the FAB part of the antibody structure. And the disulfide bridge, see, as you can see, between the two heavy chains there will be two disulfide bonds or linkages, but between the upper part of the heavy chain and the light chains there will be only one single disulfide bond. Okay, and what is the FC part? FC part is the constant part. Okay, so basically FC part is the region where the complement fixation occurs. Okay, Now again talking about the FAB region, so it is the variable region of the antibody and it is the tip of the antibody, as we can see, just the uppermost part. Okay, and it also obviously it will go and bind to the antigen, that is the already in the name only it's there, antigen binding part. And there will be specificity of antigen binding which is determined by the VH and the VL, means the variable region of the heavy chain and the light chain. Okay, and FC region will be the constant region, so it has nothing to do with the variable region. It is only present in the constant region, that is the below part, not the upper part, the lower part of the Y structure, and it is the base of the antibody, obviously, because this is the base of the Y-shaped structure, so this will be the base of the antibody. And the FC region, as I already mentioned, it will bind with the complement factors, the complement proteins, as well as the cell receptors. So this is basically a table which explains uh, some of the major differences between the innate immunity and the adaptive immunity. So innate immunity is something which is present since birth, and adaptive immunity or acquired immunity is something which is gradually developing throughout our lifetime, because obviously when a person is born, when a newborn baby is born, all the antibodies are not there inside his or her body. Okay, and also, like, forget about antibodies, the overall immune system is not developed. So gradually as the boy or the girl child will start developing both physically and mentally, because when a baby is born, the liver is very weak, the liver is not totally formed. And if the liver is one of the vital organs of the body, so if the liver is not fully formed, then the immune system will not be able to function at its full. Okay, so it's very important that the body is developed or the body is matured for the immune system to be totally active. Okay, and another important factor will be that in innate immunity or innate response, there will be no time lag. So it's an instant response, but the effect will also be short lasting, not long lasting. So it will be quick and short response. But the adaptive immunity will take a amount of time, it will take a certain amount of time to be exerted, so there will be a lag period. Lag means, as we also know in case of bacterial growth curve, those of you who have already studied, you see in the bacterial growth curve it's a S-shaped curve, and in the beginning of the, like, sigmoid curve, and in the beginning of the curve or the graph, if you see, there is a lag. So lag means something which is lagging behind. Okay, so lag period will be there in adaptive immunity because the body is taking time to respond. Okay, but the effect will be long lasting. Another important property will be that the innate immunity is not antigen specific, so it's a non-specific response. So irrespective of whatever antigen has come inside your body will be going to produce a innate immune response. Okay, irrespective of no matter what antigen has come inside your body, it can be a pollen grain, it can be as simple as a mosquito bite, it can be as simple as an allergen, or it can be as complicated as a COVID virus. Okay, but in all the cases the innate immune response will be the same, so that's why it's a non-specific response. But the adaptive immunity will be very much specific. So the adaptive immunity will change depending on what type of infection you have contracted. So when you have malaria, your adaptive immunity will be different, adaptive immune response will be different. When you have COVID, your adaptive immune response will be different. When you have typhoid, your adaptive immune response will be different. So it's a very much specific response depending on what type of antigen it is. And another important property is that, or difference is that the innate immunity will have no memory, it will produce no memory, but the adaptive immunity will lead to the development of memory cells. So for the second time, when that person is again contracting the same disease, that person will be safe. That person will no longer suffer from the same disease again for the second time. Okay. So again, this is a nice diagrammatic representation of how the adaptive and innate immune system are actually reacting with each other. Okay, so if there is an infectious exposure, there will be like the person needs to be hospitalized. So either that person will die or that person will recover. If the person recovers, and again that there is a chance that that person will contract that disease for the second time. So in that case, the adaptive immunity will be there. It will be developing a specific memory. As a result, the person will no longer develop that disease. Okay. And if the infectious exposure for the first time only is contracted by the innate immunity, is not contracted, is manageable by the innate immunity, then in the first place only the person will not suffer from any disease. Okay, so that is how the innate and adaptive immunity are actually related with each other, or they are complementing each other. Okay, so I think that's it. Thank you. I hope you liked the video. Please like and share and subscribe ITLS Academy. Thank you.